We know that microglial activation contributions to Alzheimer's disease are increasingly recognized. And rather than being just an epiphenomenon of amyloid beta and tau aggregation, recent studies have shown that microglial activation actually plays a key and is in fact an upstream in the accumulation of the AD hallmark proteins, amyloid and tau. And although we know that the amyloid beta can trigger microglial activity, we also know that microglial reactivity occurs very early on the disease progression and can occur independently of amyloid beta. And it's also very interesting to see that it only, when we see at neuroimaging studies, we only see that the amyloid beta pattern of accumulation and microglial activation only partially overlap in the human brain. So on this study, we based ourselves on experimental evidence, compelling experimental evidence showing that the APOEE4 or the APOE genotype modulates microglial activation in animal models. But however, this was never, it's unclear whether the APOEE4 genotype associates with microglial activation in the living human brain. So here we basically wanted to study the association of the APOEE4 genotype carrier with microglial activation. And on this study, we accessed 118 participants from the McGill trial cohort who had available at the same time point PET imaging for amyloid beta, for tau tangles, and also for microglial activation. Here we used PBR28 as the tracer for microglial activation. And what we observed was basically that the APOEE4 genotype associates with microglial activation in early Braak regions within the medial temporal cortex, independently of amyloid beta and tau pathologies. And this was very interesting, suggesting that perhaps the APOEE4 genotype can be a culprit of triggering early microglial activation in Alzheimer's disease.